All right, guys, sorry about the Zoom thing. I went all the way through the pandemic, and Zoom has never failed once, and it's definitely been down for a while. And then Brightspace, as usual, something always goes wrong with that, so sorry about that. But here's the lecture for today. No attendance today. This is Spring-2025. It's week 2. It's Embryology. Here we go. Cool gold eagle just about to grab its lunch there. Another cool. This is over January again, up at Sierra Vista. Just amazing. This is a juvenile gold eagle with you can tell because it has these white strips underneath its wings and tail. So very cool. Super close up of bald eagle, or of golden eagle, which are just a smidgen. Some say they're the same size, but some sources say they're just a little bit bigger than bald eagles. This guy almost got knocked off the cliff by a giant gold eagle. Wasn't even paying attention. So crazy. All right, we left off at the anterior visceral endoderm, so let's pick it up from there. And this was I guess even a little bit more review. Last time we talked about the blastocyst, how the blastocyst cavity formed, and then we said that the inner cell mass here differentiated into epiblast and hypoblast cells. We talked about GATA6 and FGF4 gene expression to cause that. We still haven't implanted, but we're going to fix that problem today. But that's kind of where we are right there, and that's where we left off. An anterior visceral endoderm is really important in forming the heart and the brain. So, and even more importantly, it tells the primitive streak where not to start. Primitive streak is basically the beginning of our nervous system and our spine and everything. So it's important the primitive streak start on the caudal port and not the cranial part of the endoderm. It'll be at that point. So let's talk about these cells because they're so important. So the trick is to turn off a gene called nodal, which is a powerful gene. We'll talk about it a lot, but we need to turn that off. So what happens, some of these cranial, and now we're going to know the caudal cranial axis once these cells form. So the cranial portion of the hypoblast cells in yellow, they undergo a differentiation. And some of these cells basically morph into these AVE cells, or anterior visceral endoderm cells. How do we do that? We turn on the nodal gene, or we turn off the nodal gene. Sorry, got a little frog in my throat. So we turn off the nodal gene is how we do that. And it's easier said than done. So how do we actually turn off the nodal gene in these cells? Again, there's a gene that is on in all of these cells, and it's secreting nodal, and we need to turn just these genes off so nodal is not expressed anymore or secreted. And so the trick is the main nodal turner offer, or in more official language, the nodal antagonist. Antagonist is a blocker. It doesn't put something into motion, it blocks something. The main nodal antagonist, it's got a very strange name, Dickoff1 or DDK1 is what gets turned on. And more specifically, DDK doesn't directly block the nodal gene, but it blocks kind of the roots of the nodal gene, which is another gene called WNT, W-N-T. So DDK1 actually goes and it turns off the WNT gene, and without the WNT gene on, nodal expression stops. So really, technically, DDK1 or Dickoff1 really indirectly inhibits nodal by cutting it off at the roots, cutting off the WNT gene. That's how that works. It's so important that there's 2 other genes that will also do the same thing, just in case you have a mutation in the Dickoff1 gene. These genes will work as well. And so these genes are lefty one, sound like gangsters kind of. Lefty 1, Cerebrus 1, and DDK 1. Sound like a tough crew. But Lefty 1 and Cerebrus 1, they're kind of backup genes, and they can do the same thing. They can eliminate or turn off Wnt, which in turn turns off nodal. So to recap, DDK1, Lefty 1, Cerebrus 1 all turn off the nodal, well, turn off the Wnt gene, which in turn turns off the nodal gene. And without nodal being expressed, now the cells will become AVE cells. And more importantly, though, these nodal inhibitors will start to seep out of the cell. Where's my little cell? Here it is. They'll start to leak upwards and upwards and upwards, and they start to leak into these green cells here. The blue cells aren't affected by this, but it leaks up into these cells. And it does the same thing up there. It turns off the nodal gene. And that's the next step. So the nodal inhibitors also diffuse out of the AVE cells, move up dorsally or toward the embryonic pole. And yeah, they do the same thing. They inhibit the epiblast cells from secreting nodal. The same thing. They inhibit Wnt, and without Wnt, nodal can't turn on. And therefore, the primitive streak cannot start there at the cranial part of it'll be ectoderm at that point. Okay, so it can't start, and that's the whole purpose. If it accidentally starts there, the fetus it won't make it. So that's very, very important to survival of the embryo. All right, just again, so turned off nodal here. The nodal inhibitor soaked up here, and they turned off nodal up here, and without nodal here, primitive streak will start on the back side of these. It'll be in the blue right here. This should have been in the blue. But you'll see when we get to that, we'll explain when we start talk about the primitive streak. Okay, just another cartoon picture. This is a better picture. Now we have a sagittal view. There's the AVE cells. They soak upwards, and they inhibit nodal expression in this part of the epiblast, which will become ectoderm in the future. Right, so it's pretty straightforward, I believe. Just another look at it. Primitive streak starts caudally. Now, there's the hypoblast cells underneath. The AVE cells would be underneath this blue sheet of epiblast cells. So they're down, but the nodal inhibitors soak up, and they knock all nodal secretion out here. So it ensures that the primitive streak doesn't start where the head's supposed to be. Primitive streak starts back here. All right, so what else do AVE cells, as I mentioned, it's not, besides inhibit primitive streak formation in the wrong place, they induce the formation of the early heart. So instrumental in forming the early part of heart development. We usually, I don't know if we'll make it that far. Sometimes we do, sometimes we don't. But yeah, so that's one thing it does. They also kind of indirectly are important for part of the brain, the forebrain development, and even part of the head development, the cartilage that turns into bone, but more importantly, the forebrain. So heart formation, forebrain, is what all AVE cells do. Indirectly, I said, because the AVE cells are very important formation of something we'll look at later called the precordial plate. All right, but let's get back to our story. Our little blastocyst has been floating around in the uterine cavity. It's taking on fluid, uterine milk's keeping it alive. But now it's time to get a blood supply going. So in order to get a blood supply going, we have to get into the endometrial wall. It has to implant. And so there's a process involved in this implantation. So let's take a look at that. Here's a little story of where we've been. There's our secondary oocyte, fertilized at this point, going down. There's a morula. We dropped into the uterine cavity, and now we're going to implant. So that's where we are now. And the presence of the blastocyst floating around in the uterine cavity causes that mucus remember I said that the epithelial cells secrete mucus. There's some are ciliated and some are secretory. And some of them secrete mucus, so we have a layer of mucus, so bacteria and viruses can't invade and sperm can't get into the endometrial wall. Well, it's time to shut that off, because if mucus is covering the endometrial wall, implantation can occur. The receptors that need to bind are hidden in the mucus, so we have to turn that off. So in physiological speak, we need to turn off the gene that makes mucus, and that's the mucin 1 gene. And we're not exactly sure what the mechanism is. It's different. All the board books seem to say something different, so I'm just going to leave it out since they can't really agree on that story yet. But we do need to downregulate. What's that word mean? Downregulate means turn off the gene. So that's what we need to do next. So mucin 1, we talked about it. It's a glycoprotein that mixes with water, and it makes the endometrial lining mucousy and slippery and slimy. And we said, yep, bugs can't get into the endometrium and sperm and things like that. That all needs to be protected. But now we need to turn it off. With this gene off, some of the receptors can finally hook up. So we've turned it off now. We're not even going to talk about the process. Just the presence of blastocyst through some unknown process turns off the mucus supply. And now the receptors become exposed. So we have troponin receptor is what the blastocyst is looking for. These troponin receptors are on the endothelial cells of the endometrium. And that's what the blastocyst is looking to dock with. So what causes these troponin receptors to sprout? And this is relatively new information. So it's our friend human chorionic gonadotropin. So Syncytiotrophoblast cells, which are derived from trophoblast cells, secrete tons of this. But it turns out that trophoblast cells, which secrete EPF, they also secrete a little bit of human chorionic gonadotropin. And it seems like this acts as not a hormone in this case, but as a paracrine. And it flows out of the trophoblast cells of the blastocyst. It soaks into the endothelial cells, and they start to sprout these troponin receptors. It may have something to do with turning off the mucus as well. But for sure, we know that this HCG causes the sprouting of the troponin receptors. So here's a cartoon of one. I guess I could have put more in here, but there's one troponin receptor that is sprouted because of HCG coming out of the blastocyst. And now we have receptors, the L-selectin receptors that live on the blastocyst can bind to the troponin receptors and dock. And once that happens, we get like an endocytosis type reaction where the blastocyst literally sinks in to the endometrial wall and goes deeper and deeper. And as it does so, we get a new cell type that's going to form. But let's go through this. On day 6, trophoblast cells express L-selectin receptors. These receptors are what the blastocyst binds to, the troponin receptors of the endometrium. And yeah, that's about all we need to know. Most of the receptors pop up at where the inner cell mass is, by the embryonic pole is where most of these occur. So docking usually occurs like that. So cool. Now we have official binding of the blastocyst and the endometrium. And great. So of course, mutations of any of these structures, troponin or L-selectin, will make it very difficult to get pregnant. And mucin 1, yes, that's turned off as well. If you have a gain of function mutation in mucin 1, what's a gain of function? We've heard that word before. That's where a gene is more powerful than it should be. So if we have a gain of function mutation mucin 1, we probably can't get pregnant as well because we can never turn off the mucus. Some more fun facts. So typically about day 22 of the menstrual cycle is where this occurs, give or take a few days. And then there's only a 2 to 3 day window where the mucus stays off and implantation can occur. So getting pregnant can be rather difficult. So that's interesting. It doesn't stay on forever. BMP genes are involved in this. I used to talk about that, but that just gets way too far into the weeds. So we don't need to say anything more about that. But we do need to talk about ectopic pregnancy, because that's always on boards. And I'm sure you'll get that in your repro class as well. Normally, this implantation that we talked about happens on the back wall of the endometrium. It can happen anywhere, but it usually happens on the posterior wall. And it's fine if it happens on the lateral or the anterior wall, that's fine. But it better not happen in the fallopian tube or the cervix or even the pelvic cavity, because that's called an ectopic pregnancy, and that's always a death sentence to the embryo, and it could be big trouble for mom as well, especially if it happens in the fallopian tubes. Okay, yep, so it can plant other places, fallopian tubes, ovaries, anywhere in the pelvic cavity. So it appears that L-selectins of the blastocyst are also able to bind to another group of proteins just called integrin proteins. There's about 5 of them that these L-selectins can also bind to that happen to be on like ovarian tissues inside the fallopian tube wall. They're not the designated target, but they can still bind, sort of, not as well. But that's what happens. The L-selectins bind to integrins of non-endometrial tissue, and now you got yourself implantation going on in a place that you shouldn't. It's not designed to have it anywhere else. And that's called an ectopic pregnancy. Happens in the fallopian tube. Little blastocysts can grow and grow and grow and burst the fallopian tube, which will send mom, before it bursts, as it stretches, there's a lot of nociceptors in fallopian tubes. It will send mom to the emergency room. They'll think she's having appendicitis or problem with the gallbladder, something like that. So it can be difficult. Those sympathetic afferent nerves aren't the greatest at telling the brain exactly where the pain is coming from. But anyway, that's what an ectopic pregnancy is. There is questions on the test about this, not the exact numbers, but you have to have an idea. For example, 96%, by far the most common place for an ectopic pregnancy to happen is in the fallopian tube itself. It can happen rarely on the ovary if the fertilized egg gets out, or if the fimbriae missed the egg and it's floating around here, or it lands on the ovary itself. The sperm, like hound dogs, those sperm can go right out the end of the fallopian tube and chase it into the pelvic cavity. So it can happen on the ovary, sometimes, very rarely, in the cervix. Hard to get through a mucus plug that forms right here in the internal os. Even more rarely on the intestines. The rectouterine pouch of Douglas, about 1%, pretty common because that's the lowest point where fluid collects in the pelvic cavity. So those are all the possible sites. All right, let's meet a new tissue. It's time for a new tissue. So we've implanted, and by an endocytosis process, the blastocyst is starting to burrow into the wall of the endometrium. And that stimulates a change. The trophoblast cells, the green cells are trophoblast cells, where the contact is with the endometrium here. A brand new cell type is formed. These green cells all split and go through mitosis. They create a new green cell, and they create a new, I don't know what color that is, light green cell. They split again, and they split again through mitosis. They always recreate themselves, but they create this new weird cell, and that's called a Syncytiotrophoblast cell. Very, very strange. Ultimately becomes part of the placenta. But as you can see here, these cells, there's 5 cells right here, and they don't have a nuclear membrane. They tend to share the nuclei with each other without a nuclear membrane. So very, very strange. and they divide so fast that they've been compared with a highly aggressive cancer. There's similarities between the two. And as they grow, it's believed that they pull the blastocysts deeper and deeper and deeper into the endometrium. This is the uterine milk out here, the uterine cavity. So they start to pull it and drag it in deeper and deeper. And you can see this maybe 6 hours or a day later, you can see it's been dragged in. Now maybe about half of it is sticking out in the uterine cavity. The rest of it has been dragged in. And that goes on and on, like this Syncytiotrophoblast dragging this big boulder through the endometrial lining. Okay, let's see. I don't think we need to say anything more. I think we've said it all already. There is a name change that embryologists aren't very good at remembering, but officially, once the trophoblast cells give rise to this new cell type, they get a name change, and that's called trophoblast. So they go from Syncytiotrophoblast to trophoblast. They're the same darn cells. They just get a name cell or a name change that tells us Syncytiotrophoblasts have been born. All right, yep, rant. When I put this class together, trying to read all these embryology books, it was very confusing when they kept switching back and forth between trophoblasts and cytotrophoblasts. But there you go. I saved you hundreds of hours of research right there. All right, in this cartoon, we can see there's the uterine milk. The blastocyst is almost completely pulled in. Very cool. Now, does anybody see a problem going on right now with regard to nutrition? Because before we had uterine milk soaking in here, and now we're going away from the uterine milk, and our little human here is going to run out of gas. It's going to need some kind of glucose, proteins, fatty acids, something to keep alive. But these are mom's spiral arteries. Sometimes they're just called endometrial arteries. So the next process we'll talk about is how mom's Syncytiotrophoblast is like a vampire. It's going to find these spiral arteries and tap into them and start sucking the blood and pooling the blood into these things called lacunae. And then we have to build a circulatory system, what we'll talk about probably next week. How does it get through? So some of you wonder, well, how does it, the Syncytiotrophoblast pulls it through, but we need to soften up the layers of the endometrium. And the Syncytiotrophoblast cells, they also secrete some things called matrix metalloprotease 2 and matrix metalloprotease 9, the MMPs 2 and 9, similar to the ones that cause degenerative disc disease like stromalase and all that gang. Very similar. So they're powerful proteolytic enzymes, and they cut their way between the endothelial cells and any fibroblasts or anything they need. They can start to kind of loosen up the tissue so Syncytiotrophoblasts can easily drag this big old thing. Well, it's not big, right? By this point, it's about not the point of the pin, it's the head of a pin. It's gotten a little bit bigger, but that's part of the game here. So MMPs 2 and 9 help this evasion. They're released by Syncytiotrophoblasts. Great. Yep, we said this. So as the blastocyst gets deeper and deeper, gets closer and closer, needs a blood supply. And yep, we talked about that. It's leaving the uterine milk, and so it's going to tap into these spiral arteries right here. And we'd be talking a lot about that. But that's one thing on its mind. It's like, how am I going to keep the little human? And there's the little bilaminar disc right there. AVE cells are not drawn in there. All right, so what else can the Syncytiotrophoblast be used for? It secretes, again, human chorionic gonadotropin, which makes sense because trophoblasts started secreting HCG. Now this one really secretes HCG in high levels, and so much so that probably by about week 2 after fertilization occurs, you can detect pregnancy. The HCG It's a hormone. It gets into the bloodstream, goes through the spiral arteries, gets in the bloodstream, and it can be detected. And that is what's used about the second week of life for pregnancy test. Remember, we said you could use EPF, which is also secreted from trophoblast cells, but that's about a $10,000 test. So what else, or what does HCG do? We talked about that. It binds to the corpus luteum and keeps that alive. So awesome. We talked about that. So it extends the life of the corpus luteum. We need the corpus luteum if you're pregnant because corpus luteum will secrete progesterone and estrogen. Those are the hormones that keep the pregnancy rolling. And we need that to go for a couple weeks or a couple months before the embryo is able to, or before the placenta is able to take over those duties. Yep, recall placenta will eventually take over secretion of estrogen, progesterone around week 12 or so, and then the corpus luteum will finally die. Remember, corpus luteum was a remnant of the Graafian follicle. So we've looked at this several times. I showed you a lecture. You can watch this. You're supposed to get this in repro, but I'm not sure if that happens. But anyway, so this is a group of 10 to 20 racers. The research varies on that. But there's a group of primary follicles that wake up. And for 2 months, they race and race and race. And one of them will become big enough to be the Graafian follicle, the winner here. Graafian follicle is the one that releases the secondary oocyte, which is stuck in meiosis II. But this goes on to be the corpus luteum because it gets stimulated by HCG to keep it alive. And corpus luteum secretes tons of the pregnancy hormones, progesterone and estrogen, to keep that thing rolling as well. Morning sickness. So there's a bunch of things that can cause morning sickness, but the number one culprit seems to be the huge increase in HCG that happens. It starts around week 2 or so when it becomes detectable. Some women are affected. My wife wasn't affected at all. She didn't have any type of nausea or anything. But all of my daughters, I have 4 daughters, female daughters. 3 of them have had kids. And they were all not feeling so good. So, yeah, HCG is one of them. The high levels of estrogen, progesterone that are secreted from the corpus luteum may have something to do with it as well. But HCG seems to be the main culprit. So what else? So why doesn't the blastocyst get attacked by mom's immune system like crazy? Because now it's really, it's inside. It's mom's spiral arteries and capillaries are right there. So he said that early pregnancy factor secretes some things that grants it immunoprivilege. Recall Syncytiotrophoblast cells? Well, they were trophoblast cells when they started, but they secreted EPF, early pregnancy factor, which granted mom's blastocyst, or which granted the blastocyst immune privilege, so mom's immune system wouldn't attack. But now the Syncytiotrophoblast does the same thing. It also helps protect. Is that slide gone? Well, I guess I won't test you on it, but it does. The Syncytiotrophoblast also grants it immunoprivilege. Maybe that slide's coming up in the next lecture because we're getting toward the end here. So another thing that happens, the next big thing that happens is we get a new cavity called the amniotic cavity and amniotic fluid. You've probably heard of that before. So that's going to happen. Let's look at the picture and see what happens. So we're almost completely implanted now. We haven't tapped into the blood vessels yet, but you can see in these epiblast cells, we've got a cavity forming right here. And that's going to be important. That's going to be called the amniotic cavity, and that's going to fill with amniotic fluid. Next thing that happens is we have to get a new layer of cells surrounding that called the amnion. But this amniotic cavity becomes massive, and this is the amniotic cavity, and it surrounds the fetus at this point, and the amniotic fluid protects it. And yeah, so that's a very important structure that we're like a day 6, day 7 or so, and it's formed. So that's important. And the amnion goes on to be part of the fetal placenta. All right, so immediately after this cavity forms, we need to make an amniotic fluid, but we don't have cells to do it, so we need to make amnion cells. And now it's time for the epiblast cells to, by mitosis, divide. So the ones on the end divide, and they create a new cell called the amnion in red. And when a cell divides, you create 2 cells. You make a copy of yourself, and then you make a copy of, kind of morphs into something else. And that repeats and repeats and repeats, and we get a new lining of cells called the amnion. And the amnion cells will eventually surround the amniotic cavity, and they will secrete amniotic fluid. And down the road, you can see when folding occurs how this gigantic amniotic cavity occurs, or how it progresses. It starts from that, and it becomes that. So, pretty cool structure there. Amnion is important. Amniotic fluid is so important to life that it's considered, the amnion is considered one of the 4 extraembryonic mesoderms. So, or extraembryonic membranes, not mesoderms. So, very important membrane. Okay, we said that already. Amniotic fluid feeds and protects. Great, there's just another picture of it. Now we can see the amnion has formed. It's already starting to secrete fluid. We can see that our cytotrophoblast, or our blastocyst, has been pulled completely within the endometrium. There's a uterine cavity out here, and uterine milk is all done. So great. That's all I got for this week. Plenty of slides for this week. There's a spotted towhee to say goodbye to you, and we'll see you next week. Email me any questions you may have. Have a good weekend.